Hello everyone, so my name is Mamiko and I'm from College of Hantek. And this summer and last academic term, I had an opportunity to work as a fellow student in Dr. Sandrigo's lab. And in the legal lab, we study the mechanisms of wound healing and regeneration of tissues using zebrafish model. We are particularly interested in how the somatosensory axons regenerate and what roles they play in tissue regeneration and wound healing. Before I start my presentation, I would like to show you what an axon is. An axon is this tube-like figure in neuron which transmits electric impulses from a neuron to neuron. As many of you know, neurons are important for us to sense and transmit information in our body. However, you might be wondering why we study neurons or axons to study regeneration of tissues or wound healing. This is because our body, when it's injured, hyperinnervates or grows excessive axons in the skin layers. And it seems the skin cells and axons somehow communicate with each other, and axons assist skins to heal or regenerate. But conversely, the injuries to the skins promote the growth of the axons. So another question is, how do the skin cells and axons communicate with each other? What is the relationship between them? To answer this question, one thing we can look at is hydrogen peroxide. Many of you might have used hydrogen peroxide to bleach your hair or disinfect your wound sites. However, in our cells, hydrogen peroxide can work as a secondary messengers. And Dr. Rieger has discovered that hydrogen peroxide is actually necessary for axons to regenerate. So in the video, you can see the axons that is marked with the green fluorescent protein grows exponentially with the presence of hydrogen peroxide after the amputation of the fin. But this growth does not happen without the presence of hydrogen peroxide. So now the question is why the hydrogen peroxide is so important for the regeneration of axons. One thing that hydrogen peroxide does in our body is to promote the formation of the disulfide bond. Hydrogen peroxide specifically targets the amino acid cysteine of a protein, and the formation of the disulfide bond between amino acid and amino acid changes the conformation of the protein. And since the function of the protein is highly dependent on its shape, this process can either activate or deactivate a protein. Another thing that is important in axon regeneration is epidermal growth factor receptor, or EGFR. And this is a receptor that is important in development. Dr. Rieger in the past has shown that the epidermal growth factor receptor is necessary for axon regeneration as well, because when EGFR is pharmacologically blocked, we do not see the regeneration of axons either. So in the video, you can see the axons, again marked with the green fluorescent protein, do not regenerate when it's blocked with different inhibitors, with the inhibitor. Also in the graph, you can see the fish compared to the fish treated with DMSO, which is a control, the fish that were treated with different inhibitors do not regenerate as much as should. So now I have told you that both hydrogen peroxide and epidermal growth factor receptor are important for regeneration of axons. Now the question is if there is a link between them. In a recent study in human carcinoma cells, it has been shown that epidermal growth factor receptor in fact has a cysteine residue that is oxidized by hydrogen peroxide. And the oxidation of this residue leads to the phosphorylation of tyrosine kinase domain and activation of the whole receptor. So now we know the link between the EGFR and hydrogen peroxide, and now the question is if it's related or is necessary for the axon regeneration. And our hypothesis is that the oxidation of cysteine residue of EGFR is necessary for the axon regeneration. To test this hypothesis, we created transgenes with wild-type and mutated EGFRs. And in the mutated EGFR, we substituted the cysteine residue, the alanine, so that the receptor cannot be oxidized anymore. So in the end, we are hoping to have 4 different constructs with wild-type and mutated EGFRs, and also with 2 different promoters. We are interested in using 2 different promoters so that we can express these EGFRs into 2 different cell types, 1 in skin cells and 2 in neurons. This is important in our study because if the oxidation of the EGFR is necessary for the axon regeneration, we would like to know if the oxidation happens in the skin cells or in neurons, and if it affects the regeneration of axons. In the first half, so right now we have the top two constructs, and in the first half of the summer, I worked on the cloning of the top wild-type constructs of EGFR. What I did was I first isolated the EGFR and promoter from the plasmid using restriction enzymes, and using an enzyme called ligase, I put them together. So now we have these constructs that we can test the activity. Then we created these two constructs, and to test the activity of the constructs, I first we transfected them into human keratinocytes. And we then performed the immunohistochemistry with anti-EGFR antibodies. And in this picture, you can see the EGFR is marked with green fluorescent protein. And as you can see, in the keratinocytes transfected with human keratinocytes transfected with wild-type EGFRs, you can see intense green fluorescence between the cells, indicating that these wild-type EGFRs are strongly located in the plasma membrane of the cells. Next, we injected these constructs into zebrafish embryos, and our initial goal was to perform the in vivo imaging to see the axon regeneration in the zebrafish. However, right now I'm facing two challenges, and I haven't really been able to do that. But right now, one of the challenges is that I observed the high mortality within the fish that are injected with mutated EGFR. And one time I observed 90% of the fish died one day after the injection. Also, I see both the cells that express the EGFR mutated and also the wild-type EGFRs in the zebrafish embryos are somehow get extruded 2 days after the fertilization and injection. So right now we are trying to optimize the concentration and the volume of the injection solution so that we can hopefully get the persistent expression and wait the fish to develop up to the optimal age for the in vivo imaging. So to summarize my talk, we have learned mainly three things. One is that both hydrogen peroxide and EGFR are important for axon regeneration. And second, both the wild-type EGFR and mutated EGFR the constructs we made are expressed by the human keratinocytes, and with the wild-type EGFR, we see the strong expression in plasma membrane. And third, we observed the injection of mutated EGFR causes high mortality to zebrafish embryos, and both the cells that express the wild-type and mutated EGFRs somehow get extruded by the embryos as they develop. So as you can see, I haven't really been able to answer the questions we initially had or test the hypothesis yet. So we will continue working on this project. But right now we know we would like to perform 4 different experiments in near future. And one of them is that we would like to detect the phosphorylation of tyrosine kinase domain and in EGFRs by transfecting the human keratinocytes and performing the Western blotting. We tried this detection with immunohistochemistry, but the antibodies didn't work, so we are trying the Western blot next time. We'd also like to perform the qPCR of ARC, which is one of the downstream targets circled in the diagram. And these 2 experiments will help us to see if the mutation and overexpression of the EGFR, overexpression of wild-type EGFR affect the functionality of the receptors. We would also like to clone the EGFRs into a neuron-specific promoter so that we can answer the question of cell type specificity. And at last, we would like to perform the in vivo imaging, and this will help in vivo imaging of axon regeneration in zebrafish, and this will help us see if the mutation and overexpression of EGFR will affect the regeneration of axons. So I would like to thank the main brief for funding me to be here, and my PI, Dr. Sandra Rieger, for accepting me as a fellow student. I would also like to thank the RAs and fellow students in our lab for their help and support. And I would like to thank the Montessori Biological Laboratory for providing me this great opportunity. And thank you for listening, and I can take now any questions.
I'm just wondering if the oxidized site on EGFR, is that required for its function?
So the question is, the oxidation of the EGFR is required for the function. So it is shown that the oxidation of, in human carcinoma cells, it has been shown that this oxidation is required because for the phosphorylation of tyrosine kinase domain and later in the activation of the receptor. But we do not know if it's necessary for the regeneration of axons yet.
I was also wondering if you were able to test in your skin cells that you were able to transfect whether when you express that mutant, if it inhibited EGR activity.
Could you rephrase the question?
When you express, you were able to express your EGFR mutant in the skin cells, and were you able to do any experiments to show that it was sufficient at inhibiting the activity in the skin cells?
So the question was if I could see if the mutation of transfection of mutated EGFR in carcinocytes showed that the function of the receptor is inhibited by the mutation. So I could not really test that yet because I haven't really been able to see the detective phosphorylation or the I haven't really see the downstream effect of the mutation. The only thing that I have learned so far from these experiments is that we see different localization of these receptors between the wild type and mutated in keratinocytes.
Any other questions? I'll ask one last question then. Sorry.
Yes.
Thinking upstream of the EGF receptor, with these mutated forms of the receptor, is there any idea that there are different stimulatory molecules, ligands, that bind to these receptors that cause the downstream effects that you see? Okay,
so I did not explain it, but EGFR receptor is actually activated. Sorry, I'll repeat the question first. The question is if there is an upstream mechanism that or ligand that binds to the EGFR receptor that EGFR receptor and that causes that works with the EGFR receptor. And then to answer the question, I didn't explain it, but EGFR receptor is actually activated by. So there are two different two same subunits, and then they homodimerize it together, and this will enable the receptor to accept a ligand, which is EGF or epidermal growth factor, and this will then upon this binding the mechanism that I explained happens, and then the downstream, and then we see the downstream effects of the epidermal growth factor receptor.
Thank you.
Thank you.
Thanks, everybody.